Okay, hi, welcome back to part 3 of chapter 17. And we just got done talking about the anatomical structure of the external, middle, and inner ears. So now we're going to go over the functionality of our ears. So let's talk about the equilibrium functionality first. Now, the hair cells within the semicircular ducts becomes active during rotational movement. So we're looking at these semicircular ducts here. And when you move your head, the hair cells within these semicircular ducts, specifically in these expanded region right here, will provide the electrical signal that will indicate how fast and in which direction your movement is. And inside of each semicircular canal is a structure called ampulla. So that's what these purple things are in this expanded region of the semicircular canals are, signifying is the region that's called the ampulla. And within the ampulla, there are specific regions called the ampullary crest. So this is the ampullary crest, which contains hair cells, these purple guys, little cells right here. And the extensions of these hair cells are embedded within this gelatinous structure here called the ampullary cupola. Okay, so ampullary crest, hair cells, and these are their extensions that extends into the gelatinous mass purple stuff right here called the ampullary cupola. So within all the semicircular canals, there is a fluid called endolymph. Okay, so this blue area, you see all this blue stuff that you're seeing all around the entire area. Those are the endolymph that I was talking about that is contained within the membranous labyrinth of your inner ears. So, this endolymph will flow in different direction when we move, okay? Which will when we will move these endolymph. These endolymph will then move the ampullary cupola, because this is the fluid around it. When it moves, it also moves this jello matter, which will then allow these hair cells to move when the gel moves. When these hair cells move, that's what's gonna bring that electrical signal towards, you see these sensory nerve right here attached to the hair cells. So it's detecting the movement of these hairs in electrical impulses as sensory information going to your brain to sense your rotational movement, specifically from the ampulla. So those hair cells within the ampulla or ampullary cupola have extensions called stereocilia and a single kinocilium. Okay, so these are all right here, stereocilia and one kinocilium. Now, these are not like the cilia we were talking about in previous chapters, about how cilia move fluids around the cells, like those lining your mucous membrane, respiratory tracts, or your fallopian tubes. Is not the same type, okay? These are like long microvilli, if anything, that are there to sense movement, okay? They're not there to move fluid. They're there to sense movement, which will then send the electrical signal to your sensory neuron that there's movement going on. So when the external forces pushes against any of these processes, it will cause a distortion of the plasma membrane that will alter the plasma membrane right here if they move, okay? And they will alter the rate at which this hair cell right here releases neurotransmitter to the sensory nerve right here that is synapsing with the hair cells, okay? So that's how we are able to generate action potential that will relay the signal for a mechanical stimulus coming from these movements of the stereocilia or the kinocilium. Now, when your head rotates, the movement of endolymph in the semicircular ducts is what will push the ampullary cupola to the side and distort those receptor processes, your kinocilia and sterocilia within those hair cells, okay? So ampulla is mainly a dynamic equilibrium structure. So I would write somewhere here, ampulla, dynamic equilibrium, which is then responsible for rotational or detecting rotational or angular movement. So when you're doing horizontal rotations, like shaking your head when you're saying no, that will stimulate hair cells of your lateral semicircular duct. And this is why we have 3 semicircular ducts. It's because we have 3 axes of movement when we talk about types of rotational movements we can do. Nodding yes in motion will excite hair cells of the anterior semicircular duct. And tilting your head side to side, like doing your lateral flexion movement, will excite the hair cells of your posterior semicircular duct. Okay? So nodding yes would be like doing flexion extension of your neck, and then horizontal rotation will be doing those lateral rotation or medial rotation about your neck. That will activate these different semicircular ducts to sense what your body is doing. Now for sensing, the portion of the endolymphic duct here, right here, is what's going to secrete. So this is endolymphic duct right here. This is what's going to secrete fluid into the endolymph, into the utricle, sacuole, and also the semicircular duct. They will secrete that endolymph continuously and allow that excess fluid to return to general location via your endolymphic sac. So a lot of times when we talk about fluid that is circulating within an enclosed space, there always needs to be a place for secretion, and there will always be a place for drainage. So where endolymph are formed is the endolymphic duct. Where endolymph are reabsorbed and returned to general circulation is endolymphatic sac. All right. So for sensing linear movements, we have a different structure called the macula that exists for static equilibrium. Okay, so macula, static equilibrium, which senses those linear movement. Okay, within the utricle and the sacuole, so this is the utricle, this is the sacuole. Within there, you see that there will be hair cells that provide the sensation of linear movement. Okay, so here is the macula. This oval circular structure here is what the macula is, and they have those hair cells that will provide those position and linear sensations. Okay, and the macula in the utricle is sensitive to your changes in horizontal movement. Okay, so macula of the utricle, which is this one right here, is sensitive to changes in horizontal movement, whereas macula of the sacuole is sensitive to changes in vertical movement. So that's this one right there. Okay, that is sensitive for vertical. This one is sensitive for horizontal. Okay, now and just like the ampullae, okay, the hair processes are also embedded within another gelatinous structure, but is not called the ampulla cupola. Okay, it is not the ampullary cupola. It is called the otolithic membrane. Okay, so this whole blue region right here is the gelatinous matter that exists on the macula. Okay, that is for moving those hair cells within those stereocilia of hair cells of the macula. Now, on the surface of the otolithic membrane, there are these purple crystals here that you can see in this image. They are actually calcium carbonate crystals called otoliths, and they're also known as ear stones by common name, but they are the ones that's going to be moving around, which then will move the otolithic membrane around, which then the otolithic membrane will cause movement of these stereocilia to send that sensation of movement in these nerve fibers here to give you the sensation of linear movements. So when your head is upright, the otolith is sitting right on top of the otolithic membrane right there. So it compresses, and the way it kind of presses on these stereocilia of the hair cells on macular surface, which then tells your brain that your head is upright. Okay. Now, when your head is tilted posteriorly, gravity will actually pull the otolith backwards, okay? And that will distort the hair cells, stereocilia, in that same direction that you're tilting. And this change in receptor activity is what tells your central nervous system that your head is not level, and you know and understand that your neck and your head is tilting backwards. Now, tilting motion and versus linear acceleration is two different things. Now, the same thing can be used for linear movement. This is talking about tilting, this moving backwards. But if this was on a level surface, okay, for example, a level surface of the otolithic membrane, and this is the receptor hair cells, okay, within the macula, and here are those stereocilia sticking out, and there's that crystal sitting on top, otolith sitting on top of the otolithic membrane. So let's say you're in a car, and all of a sudden the car accelerates very fast. So that linear acceleration is going to cause the otolith to go backwards, which makes you feel that you're moving in backwards, push against the back of your car seat and moving forward. Whereas if, let's say, the driver decides to suddenly stomp on the brake and you move forward instead, and the otolith then also gets moved forward, which tells you that your body is moving and you realize that you are moving forward. So that's how linear movement are sensed in that similar manner. It's just that the otoliths are moving in a linear back and forth situation versus being tilted back and forth situation. Okay? Now, tilting motion versus linear motion can be distinguished by your brain due to the fact that they're integrating both vestibular sensation with visual information. So the fact that you're seeing that now you're looking forward and then all of a sudden you're looking at the ceiling tells your brain that you're tilting and not doing linear acceleration. Or when you're in a car, you see, moving environment past you. So that together with your vestibular sensation is what tells you that you're accelerating forward, moving forward. So hair cells of the vestibule and the semicircular canals are monitored by sensory neurons within the vestibular ganglia. So here's your vestibule. Here's the vestibular ganglion right there, which will then form sensory fibers that's going to travel through the vestibular nerve. Remember, you have two divisions of the cranial nerve 8. Cranial nerve 8 is called the vestibulocochlear nerve. So that nerve actually splits into two divisions. One goes towards the vestibule, one goes towards the cochlea. Okay, so for balance, this vestibular nerve will send information towards the vestibulocochlear nerve, and the vestibulocochlear nerve, which is cranial nerve 8 of your brain will then innervate neurons within the vestibular nuclei. Okay, right here, this red thing. And the vestibular nuclei actually sits between your pons area and your medulla oblongata. Okay, so this vestibular nuclei. And that's how we can sense those balance and equilibrium from our either macula or your ampulla. Remember, ampulla is for rotational movement, dynamic equilibrium. Macula is for static equilibrium, sensing linear movement. So now, if the brainstem or the internal ear is damaged, because technically speaking, this is within that brainstem area, or if any of these nerves become damaged, or even the vestibule itself becoming damaged, your automatic eye movement will occur, okay? Because technically speaking, how your eye move have to do with your sense of equilibrium and balance. And that's why in lab, when you did those tests, your bearing knee tests, when you're sitting on that rotating chair, when you're rotating, your eyes will move in response in the direction of your rotation, because your semicircular canal are working properly to sense that you're turning in rotation. And that's why your eyes end up moving. But that eyes moving back and forth is called nystagmus. Now, nystagmus is normal when you are rotating and doing the movement. But for some people, it actually exists while they're being static. Okay, they're just sitting there and standing up, but yet their eyes are in that constant movement. Okay, so that's nystagmus present in a normal person that is just standing still or sitting still, which is an abnormal situation, which can be indication of the vestibule of the inner ear not working, or somewhere within the brainstem is damaged. So they are constantly experiencing nystagmus when their body is stationary. Okay, so they have trouble controlling that eye movement. And before I get to the auditory part of the ear, I'll show you this quick video that shows you the animation of these equilibrium sensing structures in action.
The human ear is divided into 3 compartments: the external ear, middle ear, and inner ear. The inner ear contains the spiral-shaped cochlea, where sound waves are transduced into neural signals, and the vestibular complex, which contains the receptors for our sense of equilibrium. The central, egg-shaped cavity is the vestibule, which contains a pair of membranous sacs, the saccule and the utricle. Inside the utricle and saccule are hair cells similar to those in the organ of Corti. The hairs are clustered in the macula, where their processes are embedded in a gelatinous mass and lie under a thin layer of crystals called otoliths. When the head tilts, gravity moves the crystal mass and distorts the stereocilia of the hair cells. This is how the saccule and utricle provide information about position with respect to gravity. Behind the vestibule is the third portion of the bony labyrinth, known as the semicircular canals. The canals project from the posterior region of the vestibule and are responsible for the detection of head motion in 3 spatial planes. The anterior duct senses forward and backward motion. The posterior duct detects moving up and down. The lateral duct senses moving left to right. Each canal contains a membranous semicircular duct where angular momentum is sensed. At the base of each duct is an expansion called the ampulla. Within the ampulla, long stereocilia of hair cells are embedded in the cupula, which sticks out into the endolymph. When your head moves, the endolymph moves the cupula and stimulates the stereocilia.
Okay, so that wraps up, wraps it up all in one nutshell with the action. Oh, we don't need to watch that again. All right, so now on to the auditory sensation part of the inner ear, which is made possible by the structure called the cochlea, which is this nail-shaped organ right here, snail shell shaped organ. Within the cochlea, is really one long tube that is curled into that shape. Okay, so it kind of goes from here and then it goes into that shape to form that spiral looking thing. So if we were to unroll this spiral looking thing outwards, we will have one long tube going this way. Okay, we'll have one long tube going this way. And again, because we need to save space in our body, everything within our body is all about compaction. So instead of making a long thing that extends into the skull and taking up all your room, we end up rolling it like this to conserve space and make sure things are compact. So if we unroll it okay. So we unroll it, we'll see that the tube actually contains something that looks like this. So if we unroll it and then cut a cross section of it, you will see that the long tube is divided into 3 different hollow ducts. Okay, this one right here is called the cochlear duct. Okay, this one right here is called the vestibular duct or scala vestibuli. Okay, and then this one right here is called the tympanic duct or also known as scala tympani. The cochlear duct is also known as scala media, but I find that the scala is kind of hard to remember. So I like to just remember as cochlear duct, vestibular duct, and tympanic duct. Okay, and how I know that this is the vestibular duct is because I see that there is the vestibular membrane right there. If you look at it, there's this diagonal line going this way that is absent on this part of the duct. So that's why this is the vestibular duct being right next to this diagonal vestibular membrane. The one in the middle is always the cochlear duct, and then the one on the bottom without that diagonal thing of vestibular duct is then the tympanic duct. Make sure you don't mistake tympanic duct with tympanic membrane. Tympanic membrane is your eardrum. This is a tympanic duct that's within the cochlea itself. All right, and the hair cells that are responsible for sending electrical impulses resides in a structure here called the organ of Corti. So we zoom in to that region. This is how that area looks like, and on the microscope, this is how it looks like in your lab exercise, for example. So this organ of Corti sits on another membrane called the basilar membrane, which is this one right here. Okay, this blue thing right here on the bottom is called the basilar membrane, and it is what separates the cochlear duct from your tympanic duct. So cochlear duct, tympanic duct, bottom right here of the organ of Corti is the basilar membrane that will vibrate, which will cause the hair cells to push against the tectorial membrane. Again, these hair cells is what's going to tell you those electrical impulses to get to your brain to tell you that you are sensing sound. So like I was saying, the hair cells are usually arranged in series of longitudinal rows, and their little stereocilia right here are sticking up, hitting next, kind of close to the tectorial membrane. This part is the tectorial membrane, just like how this part is the tectorial membrane. And when the basilar membrane kind of vibrates up and down like this, it's going to push the hair cells against the tectorial membrane, which will then move these cilia out of position, stereocilia out of position, and that will cause an action potential and electrical impulse to be propagated down your cochlea nerve for sensation of sound. Now, within the tectorial membrane, those hair cells do not have kinocilia. That's only present for the ampulla region. All right. So here's the final story of the chapter, which is how hearing is perceived. So make sure you pay attention to how hearing is perceived. So you have sound waves coming in from your auricle, directed towards your auricle into your external acoustic meatus or your auditory canal. That frequency of sound wave now is going to hit your tympanic membrane right here. And tympanic membrane will vibrate in resonance, which means in the same frequency and same amplitude to the sound wave, the original sound wave that's coming through. So it's going to vibrate just in that same frequency and amplitude, and that will relay that same vibration to your three auditory ossicle bone, which is first to the malleus, then to the incus, then to the stapes. The stapes, being vibrating this way, will then push this particular oval window, because that's what the stapes is attached to, is this oval window. Well, the oval window is attached to this part of the cochlea. This is actually the cochlea that, spiral around, okay? And within the cochlea, you have the bony labyrinth versus the membranous labyrinth, okay? So these are those ducts we were just talking about that I'm going to get to, okay? So this is how sound is relayed from the external area to the middle ear, and then it will be relayed to the inner ear. And once your stapes move inward, if it move inward, there's fluid here that's going to go through the entire cochlea and then comes back here to move this round window outwards. Okay, so this round window here outwards. So when this moves in, this round window will move out. Okay, and when this moves out, this round window will move in. Okay, so it's always these two here moving in opposite direction. Okay, in class when I see my students in person and I get to do that oscillating motion, it's hard for me I don't have two pointers here to do it at the same time, but these two, oval window, round window, are doing opposite motion. When oval window pushes in, round window pushes out. When oval window goes out, round window goes in. Okay, so that is what's causing the same vibration that's relayed within the perilymph of these two ducts. Okay, there are vestibular duct and your tympanic duct. The center here is your cochlea duct. All right, so pressure waves travel through the perilymph from your oval window towards your round window and will cause these perilymph to vibrate in that same frequency of the sound that's coming through. Now, when the sound goes, depending on the frequency of sound, they will cross over at different points of your basilar membrane. Okay, so this right here is your basilar membrane. This is your cochlea duct. Okay, you see cochlea duct, which contains endolymph. This is your tympanic duct. This is your vestibular duct that contains perilymph. So depending what frequency range that sound is coming through, if it's a lower frequency, that sound wave would travel all the way almost to the end and then cross over to the basilar membrane, vibrate the basilar membrane at that particular amplitude, and then come back outwards. Okay? But if it's a high frequency sound wave, then it's going to start turning around very quickly closer to the oval window. Okay, for example, in 16,000 hertz or 15,000 hertz, and vibrate the basilar membrane around that area closer to the oval window. Okay, so higher frequency, shorter wavelength, vibrate closer to the oval window. If it is a louder sound, it's called more vibration of the basilar membrane. If it's a softer sound, small little vibration. Okay. If it's a low frequency sound, then it will go travel all the way here and vibrate the basilar membrane here. That would tell you that what frequency you're hearing according to where on the basilar membrane along the entire cochlea tube that it is vibrating at. So this vibration of the basilar membrane will cause those hair cells that I was talking about. Remember, the organ of Corti kind of looks like this here with the tectorial membrane, and then here's that wave looking thing with the hair cells like this and the stereocilia sticking up. So the basilar membrane is down here within that organ of Corti. So basilar membrane vibrates, causing these hair cells to also vibrate. And these hair cells will cause these stereocilia to push against the tectorial membrane, which will then displace those stereocilia. And hair cell will then become depolarized, releasing neurotransmitter to your sensory neuron. Specifically, it will go on to your cochlea nerve, which will then travel towards your brain, which is particularly the auditory cortex of your temporal lobe. Okay, so information, like I was saying, from about the region and intensity of stimulation. Region meaning, is it high intensity or is it low intensity within the basilar membrane that's stimulated, and then it will be relayed to the central nervous system over the cochlear nerve towards the medulla oblongata, which will then get to your thalamus. Then your thalamus knows that this is auditory information, which then will go towards your auditory cortex within your temporal lobe. Okay. All right. So this is a video that wraps up sort of all that I was talking about, the whole story of how sound travel from outside toward your inner ears, okay, and how we're able to perceive them.
As sound waves enter the ear, they travel through the outer ear, through the external auditory canal, and strike the eardrum, causing it to vibrate. The central part of the eardrum is connected to a small bone of the middle ear, called the malleus, or hammer. As the malleus vibrates, it transmits the sound vibrations to the other two small bones, or ossicles, of the middle ear, the incus and stapes. As the stapes moves, it pushes a structure called the oval window in and out. This action is passed on to the cochlea, which is a fluid-filled snail-like structure that contains the receptor organ for hearing. Let's unwind the cochlea to get a closer look. In this enlarged cut view, you can see the spiral organ of Corti, which is the receptor organ for hearing. It consists of tiny hair cells that translate the fluid vibrations of sounds from its surrounding ducts into electrical impulses that are carried to the brain by sensory nerves. Let's take a look at this process in action. As the stapes rocks back and forth against the oval window, it transmits pressure waves of sound through the fluid of the cochlea, sending the organ of Corti in the cochlear duct into motion. The fibers near the cochlear apex resonate to lower frequency sound, while fibers near the oval window respond to higher frequency sound.
Okay.
As sound
This wraps up chapter 17, so let's go over the practice questions now. What is unique about sense of smell when compared to other special senses? Olfactory stimulation is the only type of sensory information that reaches the cerebral cortex directly, and if there's another choice to talk about, it would be that olfactory neuron is the only one that can regenerate as a neural tissue. One of the few examples of neuronal replacement of cells in the adult, which was what I was just talking about, is the olfactory receptor population. The area in the brain that corresponds to gustation is called insula. Okay, remember Broca's has to do with words, okay, language. Occipital is the vision. Parietal is your sensory for touch, pressure, and all that stuff. A loss of transparency in the what will result in what? We cannot have loss of transparency within the lens, which will cause cataracts. What processes takes place for the lens of your eye to become rounder to focus on nearby object? What does your ciliary muscles do? They will contract, which reduces tension on the suspensory ligaments, allowing your lens to become more round shape for a close-up object. Which of the following statement applies to detached retina? All of these are correct. There's separation between the neural part of the retina and the pigmented part. That's the whole idea of detached retina. To heal a detached retina, the detached layers needs to be reattached. Oh, that's what I didn't talk about. So let me come back to talk about this. What happens with detached retina is that in order to treat it, they will use a laser that will sort of put spots around the kind of like use, if you think about how nails and screws screw things in and staple things together, that's what that laser is doing when you're trying to heal a detached retina, is that they will put the retina layer back on. So let's say if this layer fall off from the choroid, they will put this layer back on there, and then they would burn a laser beam through to glue it in a sense in multiple places to make sure the retina now sticks back on the choroid layer or the pigmented layer of the retina, but it will produce multiple more blind spot as a negative effect. But having multiple blind spot is still better than being completely blind from a detached retina or big portion of your images, not working properly. We rather afford a few more blind spots. Okay, so that's how detached retina is treated. Okay, vitamin A is used to synthesize which component of rhodopsin? Retinol. Opsin is the protein. Retinol is the vitamin part of it. Why can corneal transplant surgeries be performed between unrelated individuals without the risk of rejection? Because there are no blood vessels within the cornea, so white blood cells can't try to go there and attack the tissue. When aqueous humor cannot drain into sclerovenous sinus, what happens? You end up with glaucoma. How does vitamin A deficiency affect vision? They will decrease the amount of visual pigment because you're dependent on vitamin A to regenerate rhodopsin. And rhodopsin is an important visual pigment that is important for your photoreceptors. So you would end up with poor night vision when you don't have enough vitamin A, called nyctalopia. What is the role of auditory vascicles in hearing? They act as levers to conduct vibrations to the inner ear, so that's why they form those synovial articulations with each other. Vibrations of the what is transmitted at the what to produce a sensation of sound. First, vibration of the stapes is transmitted to the oval window for that sensation of sound. Malleus, it actually goes on the incus, and incus have to go on the stapes, and then the stapes will then touch the oval window. Round window is at the other end. If you're attending an incredibly loud rock concert, what is the reaction of your tensor tympani to the noise? Remember, this is the muscle, so they will pull the malleus medially, okay, and stiffen the tympanic membrane up so you don't end up hurting or damaging your tympanic membrane. Which structures comprise a major subdivision of the bony labyrinth? Your vestibule, 3 semicircular canal, and the cochlea. The whole bony part of your inner ear. What are the sensory receptors that respond to rotational movements of the head? Rotational movement. So it has to be your ampulla, which is within your anterior, posterior, and lateral semicircular duct. Remember, within the semicircular canal, there are those expanded regions at the end, and that's where that ampulla is for rotational movement. Low frequency sounds vibrate at the basilar membrane. Where? Because what? They vibrate far from the oval window. Remember, the cochlea kind of curves like that in that snail movement. So if we unroll this to one long tube, the lower frequency happens farther away from the oval window, whereas the more high frequency would vibrate at closer to the oval window, and they are vibrating farther is because they have a longer wavelength when you have low frequency sound. Longer wavelength, shorter wavelength. Low frequency, high frequency. Okay, all right, and that is all I have for you.